You probably know by now that plants use energy from the sun to convert carbon dioxide from the air and water from their roots into glucose. But of course, it's not that simple. Like I mentioned in the overview video, photosynthesis is actually divided into two parts, the light reaction and the Calvin cycle. The light reaction is going to address the primary challenge that plants face. Only a very small part of the entire structure can access light energy, and those parts are molecules known as pigments. In the light reaction, pigments are going to first access the energy from the sun and kick off a multi-step process that passes that energy on to the electrons of the pigment molecules, which are put to work to create our energy-carrying molecular batteries. Those batteries will be sent on to the Calvin cycle, where that stored energy will be used to convert carbon dioxide into glucose. At the end of the Calvin cycle, our depleted batteries come back to the light reaction to pick up more energy. The light reaction takes place in stacks in the chloroplast known as granum. Each stack is made up of flattened pancakes called thylakoids. For the rest of the video, we're going to look at the process that takes place in one thylakoid and show that thylakoid as a cross-section. Each thylakoid has a hollow space called a lumen, which is bound by a membrane. Like every other important membrane in a cell, that membrane is made up of a phospholipid bilayer. The outer-facing portion of the bilayer has the phosphate heads, which are hydrophilic and can mingle with water. The other part of phospholipids are the lipid tails, which are sandwiched between the two layers of phosphate heads. These tails are hydrophobic and don't like interacting with polar molecules or charged ions. Only certain substances can diffuse easily through a phospholipid bilayer, but there's plenty of stuff that can't. To help those substances move in and out of the membrane, there's stuff embedded in the phospholipid bilayers, like protein channels and various pores that provide a passageway for things like charged particles, which normally could not get through the tangle of hydrophobic tails. For photosynthesis, hydrogen ions represent the charged particles that can't get through a phospholipid bilayer on their own. They'll be important for later, so hang on to that info. So now I'm going to work on my cross-section of a thylakoid, which is contained on all sides by a phospholipid bilayer. The hydrophilic phosphate heads face both the watery environment that makes up the stroma outside the thylakoid and the lumen inside the thylakoid. The hydrophobic tails are sandwiched in the middle, where they are protected from that watery environment. Embedded in that phospholipid bilayer, there's going to be some functional stuff. First, the electron transport chain, which will later provide a passageway for hydrogen ions into the thylakoid lumen. Second, we have photosystem 2 and photosystem 1. Photosystems contain our very important pigment molecules, which again are the only molecules that can actually interface with solar energy. Solar energy from the sun in the form of photons is going to hit both photosystem 2 and photosystem 1 at the same time. For the rest of this video, I'm going to first explain what happens at photosystem 2, and then explain what happens at photosystem 1. But it's important for you to remember that these processes are happening simultaneously. When solar energy hits the individual pigment molecules, an electron becomes excited and is bounced from one pigment molecule to the next until it hits a reaction center. Once the supercharged electron hits the reaction center, it leaves photosystem 2. However, despite escaping the confines of photosystem 2, that supercharged electron is pretty immediately captured by the electron transport chain, and it puts the super solar charged electron to work by stealing that energy to pump hydrogen ions into the lumen. Now's a good time to mention something important. These videos I'm making are primarily aimed at non-major students in a general biology course. If you're an intro bio student in a 4 majors course, there's a solid chance your professor wants you to know the names for the parts of the electron transport chain. I'm simplifying here, and you should be aware of that. But anyways, the electron is passed down the electron transport chain, with more and more of its solar energy depleted to pump hydrogen ions into the lumen. However, photosystem 2 now has a problem. It's lost an electron, and that electron needs to be replaced. The electron from photosystem 2 is replaced by the breakdown of water. Water is going to split into hydrogen ions and a bit of oxygen gas, which can then diffuse out of the thylakoid, out of the chloroplast, and eventually out of the plant where we get to access it. Thanks, plants. From the splitting of water, an electron is also released and replaces the lost one from photosystem 2. At the end of this process, you've now generated a lot of hydrogen ions within the lumen. Those hydrogen ions want to flow down their concentration gradient, but they cannot diffuse through a phospholipid bilayer because of those hydrophobic tails. They need a safe passageway. This is where ATP synthase comes in, which is going to provide the only passageway that the hydrogen ions can travel through. ATP synthase is going to function like a hydroelectric dam. The hydrogen ion gradient is our dammed water, and as those ions flow through ATP synthase, they provide the energy to do work, much like water flowing through a dam can be harnessed to produce electricity. The flow of the hydrogen ions is going to allow ATP synthase to produce adenosine triphosphate. ATP synthase will take adenosine diphosphate and phosphate and attach them together. Adenosine diphosphate has 2 phosphates, you attach a third, and that's how you end up with adenosine triphosphate. This is how you create the first of your charged molecular batteries, ATP. Meanwhile, photons are hitting photosystem 1's pigments, an electron is getting excited, reaches the reaction center, and leaves photosystem 1. It's picked up by some proteins where it is also forced to do work. However, unlike the electron from photosystem 2, the electron from photosystem 1 is used to create our other charged molecular battery. I'm going to use the acronym here because it's a ridiculously complicated name, but NADP is combined with a hydrogen ion to make NADPH. Photosystem 1 needs to replace the electron that it has lost, and conveniently, it does that from the energy-drained electron that originally came from photosystem 2. So through this ridiculous process, we have converted solar energy to energized electrons, which do work to create our charged batteries, adenosine triphosphate, and NADPH. These batteries will go to the Calvin cycle and power the reaction that converts carbon dioxide to sugar. To study something like this, you want to have 2 approaches. First, make sure you can draw this process and explain what is happening at each step. Check yourself against this video or your textbook. Once you have that down, you need to pretend to be your professor and try to predict what sort of test questions they could come up with. I'm never going to ask a student to draw out the process of the light reaction, but I will ask them questions like, what does the electron from photosystem 2 do? How is the electron from photosystem 2 replaced? How is the electron from photosystem 1 replaced? What powers ATP synthase to create ATP? The best way to study for these topics isn't just to memorize the process, but also to think like a professor.